Go ahead, pilot, watch lead. Copy that, standing by.
The pilots are just getting settled here. Looks like maybe we can see a fine dusting of sediment on the rock. We're going to get a better view here in just a second.
This kind of looks broken up.
Yeah, it looks like a bit of rubble coming probably from upslope. We're expecting to see probably a fair amount of manganese crust on some of these seamounts we'll be diving in. In fact, that's one of our motivations here, hopefully to get a sample, look at the chemistry of some of these crusts, which tend to form on the sides of these seamounts. Now, they actually usually form here because we have low sediment input. That's the best sites. The currents will tend to sweep the sediment away. Here we've got a fairly good dusting.
But I also see what look like broken up pieces. I don't know if they're lodged or glued in there, and some of them look kind of angular, so
yeah,
might have landed in a spot where you can get a rock right away, perhaps.
Might get lucky.
Yeah.
So Alan and Malcolm, please chime in if you see something that's particularly of interest, and once the pilots are settled here, we'll try to guide them towards it.
I see that Astrid Leitner at University of Hawaii is also noting that a habitat like this with the dusting of sediment and so on, while we're not seeing any immediate sessile fauna, might be good for fish. So, Del, the top of this feature isn't particularly flat, so I imagine these are probably pelagic sediments coming out of the water, you think?
Yeah, that's sometimes these flat top volcanoes are that way because they've had a coral reef on them and the volcanoes have subsided. This one isn't quite flat, so it doesn't look like there is a coral complex. This would probably be pelagic sediment. And then the bigger boulder sized things we see here are coming down, probably coming down slope.
So I think if what little I know of the geology, if it is pelagic sediment, then I guess we could predict that we'd see a lot of shell bits and planktonic organism bits making up that sediment.
Yeah, I'm not sure how many shells we'd see, but yeah, certainly.
I meant like pteropod shells and foraminiferal shells, that sort of thing.
Yeah.
So we're in a process where the different vehicles are getting settled, make sure the ship's in a good position and everybody's in a good position. We're all safe before we begin our transect and start looking for things actively. It's pretty cold down here. I see the water temperature is 1.79 degrees Celsius at the bottom here at 2,480 meters, so less than 2 degrees Celsius. Copy that, pilot. I think you may have heard us remark that maybe we think some of this is broken up, so I'm going to turn it over to Del to direct you maybe to think about a rock sample.
Yeah, Alan, would this be a good place to sample from your perspective?
Yeah, I think so. I'd be inclined to try and look for a good angular piece. One of the things we have to be cognizant of is the likelihood of alteration on any what would have been fresh basalt. So a relatively fresh piece, if that's at all discernible, would be nice. A lot of the more rounded-looking rocks, I'm wondering if they don't have probably a significant manganese crust on them. So, you know, every now and then one sees some darker-looking, more angular pieces, and I'm wondering if that's maybe the best starting point for a sample. As large as is possible, the better.
Okay.
From what I understand, a good chunk of the outer part of the rock needs to be effectively thrown away for decent geochemistry to be performed on it.
Alan, this is Chris at the University of Hawaii, and it looked to me like I saw some pillows over there to the right. Would you concur, or is it just an optical illusion? Or Elaine, excuse me.
I'm having not a difficulty, but the scale is a little difficult to make out from my end. Maybe Del could give a comment as to what the size of some of those rounded features are.
Yeah.
Whether they
Pilot, could you turn on the lasers?
They're on.
Oh, they're on.
They're
Oh,
on.
I didn't see them.
See,
Okay,
right
so
there.
if you can make them out, which I apparently couldn't. They're 10 centimeters apart, so that gives you a sense of the scale there. I agree with you,
that's
Chris.
about 10.
So one of those little things that looks like a pillow, there is about 10 centimeters across. It could be these are just loose pieces, and we're getting this crust built over them, kind of gives them an appearance. But yeah, they do have that look.
On that close-up, these all look like they're pretty well set in place. Yeah, I think that might be a crinoid, perhaps, or... Yep. So we have our first verifiable biologic here. I thought I saw a sea cucumber before, but this is an Echinoderm related to a starfish, and it's called a crinoid. You can see all its arms up there coming off the rock, and the little side branches have tube feet in them, and they can pull food out of the water column, and the mouth is tucked just down there in the center where all the arms meet. Nope, I think that is okay. Thank you.
So, pilot, this is watch lead. We're going to be on the lookout for a angular rock. The bigger, the better. So, if you spy something like that, you want to head to it. That would be great.
I do see a coral up here that's just in the upper center of the screen right now. Maybe 3 branches, possibly a bamboo coral. It's very white in color. So one of the things, if this was a lot of loose rubble, you wouldn't expect to see any larger corals growing on it because they need a very firm substrate from which to grow, and if the rocks were loose, they would tumble and they would constantly be broken up. So this is a good indication perhaps that this is all pretty solidified in place and not a pile of rubble. So this is our first coral of the day. It's always nice to see corals when you first get down. It's a good sign of things, and this is indeed a bamboo coral, which is one of the more common octocorals. Octocorals are the group that includes all the sea fans, and the bamboo corals are one of the more common families of deep-sea octocorals. And you can kind of see, and you'll see later as we look towards the base, it's very white. It has a skeleton, and the flesh that is on the skeleton is transparent, which is why you can see that skeleton. And then you see all these little flower-like things that are arising from it. These are the polyps. These are the actual feeding structures. They have a ring of tentacles. And you can see in the center there, there's a little pink dot, and that's where the mouth is. And all of them are interconnected with each other, so they basically have a continuous stomach. And this entire colony arose from a single egg, a single larva that settled here and has now grown into this shape. You can see there's some little white bumps running between those different polyps. And that's a very interesting thing we've only seen, or I've only seen a few times, and only on these Okeanos dives in the Pacific. And we think they may be places where there are lots of stinging cells. And that might be some indication that there's some predator around here disturbing it. And then you can also see some spine-like structures that are between the tentacles. Those are structures called sclerites. I'll talk more about those later, but they're calcium carbonate skeletal elements that help give support or provide defense and so on, but we use them as taxonomists to identify these corals to species. As we go down here, you're going to see in a minute, there we go. So the tissue is ended, so now you get a really good view of the skeleton without tissue. So presumably something may have been feeding on this coral. And we see wrapped around the base of the coral a brittle star. We don't think these brittle stars are feeding on these corals, but we may have to revise that understanding. Maybe they're just scraping the flesh and eroding it off, but I think we've made a few observations this year where we've seen these ophiuroids with these really sharp spines on the arm, and they seem to lack tissue. I think the idea was that these sorts of animals are getting food out of the water column or other things that have landed on the coral. But one last thing I want to point out, there's a black band there between the white skeleton. That's a node, it's made of protein, and one of the reasons this is called a bamboo coral is because the skeleton reminds you of bamboo because it alternates between the white and the brown. Thank you, that was a great observation. Does anybody on shore have anything to say about this before we leave? So I don't know for certain what species of bamboo coral that is, but it did look reminiscent to something that we saw last year with those very prominent spines, and then something I was looking at was the way other sclerites were arranged in the body wall of the polyp.
Scott, this is Shirley. It looks like on these rocks there are some
Pilot.
very small, soft spheres. I don't know if that's just like, sediment or if it's something biological. Maybe you could zoom in on one of the edges of one of these rocks when you stop the next time.
Great, Shirley, will do. Thank you for the heads up on that.
Yes, some of these structures are certainly continue to look like pillows here.
So pillows, this would be an indication of a recent eruption or something?
Well, not necessarily recent, but this would indicate these are lavas that were emplaced and are more or less intact here, not rubble that has come down and then been crusted over. They could still be quite old. The sediment we see here, it may take a long time for that to accumulate.
I see an anemone down at the very bottom center of the screen. The fact that it's poking out of the sediment suggests it may be a tube anemone, what we call a serianthid. If we could get a quick zoom if we haven't left. Thank you, Chris. And I'm seeing as we're coming in, there may be one of these little stalk-like features that Shirley Pomponi from Harbor Branch was just pointing out. Just to its right, there's a white structure coming off the front face of the rock. So one of the scientists in our chat room is Tina Molodtsova from the Shirshov Institute in Russia, and she is an expert in this group of tube anemones. So I wonder if she'll have any observations to share. Well, so I said it was a tube anemone, and this is why we need to do the close-ups. This is not a tube anemone. It is a sea anemone, or I think it is. I mean, the alternative would be that it's a cup coral, and I actually can't see the cup. But as we zoom in, I don't see anything that looks like it's a calcium carbonate structure underneath it, which would suggest that this is an anemone. It may be a burrowing anemone of some kind. It does appear to be in the sediment. You can see that sediment's really coarse, isn't it?
It is, yeah.
A tube anemone is something different that builds a tube, and it would have a second ring of tentacles around the mouth. The mouth is that little slit that's in the center of those tentacles. Thank you, Pilot. That's good for us. And just as you're coming out just to the right of here, let's look at the front face of this rock. Okay, right there. Okay, that's not a sponge. It looks like maybe it's an agglutinated tube of some kind. meaning that something has glued together different bits and pieces from the sediment to make a tube, and that's the sort of thing that various worms do, or foraminiferans, which is a single-celled organism. Thank you. Shirley agrees that that is not a sponge right there anyway. And Tina Molitsova does agree that tentacles don't look like a scleractinian, she says, and a scleractinian is the formal name for the group that includes the cup corals. So I think we would all agree that that is a sea anemone that has burrowed somewhat into the sediment.
Great, thank you.
I think I see a whip. It's at about 10 o'clock to the lasers. It's really fine. It's on a row of rubble just before that sediment patch. Oh, great. This is a sponge. Shirley, are you on the line? You want to talk about this? I even see the 2 amphipods on it.
I'm on the line. I'm trying to get a focus here on what it is we're looking
Yeah.
at. We're kind of whited out here.
Yeah, it's quite small. It's between the lasers and we'll...
Yeah.
Shirley, it could be a cladorizid, but it was moving pretty quickly since I'm sure.
Yeah, we're setting it up
I'm
for another
looking
view.
now.
I'm pretty sure it's a cladorizid, which, well, I'll let you guys talk about it. Understood.
So, if in fact that's what it is, cladorizids are a particular group of demo sponges. So they make spicules, so these microscopic skeletal elements out of silica. But unlike most other sponges, they're not filter feeders. They capture their prey with their spicules, kind of like a Velcro surface. But let me, before I go into too much detail here, let's make sure that's what it is.
Yeah, I'm sure you're correct, Shirley.
Yeah. Yep, there we go. And so they have these little spicules or skeletal elements that make the surface very rough. And so very small crustaceans can get caught on the surface, and then the sponge cells completely engulf the prey that they've captured and digest it. So they're very unusual and unlike other demo sponges and other sponges in general that filter feed. And it's an adaptation for life in the deep sea where there's not a lot of particulate matter that they can filter out of the water column.
So great, sponges that have become predators. And the 2 things that you're seeing sitting on the sponge, these aren't prey. These are 2 what are called amphipods, a kind of crustacean related to the roly-polies that you may find in your basement, the little isopods. And when we have collected things like this before, we've found that one of them is a male and one of them is a female, and they're kind of specially adapted for where they are. You can see well, you may not be able to see, but they have these legs that are formed kind of like hooks, back legs, that they're used to hold on to the main stem of the sponge, and they have these very long antennae that go out in the water, and I suspect they're kind of sniffing the water for food. Thank you, Pilot. That's terrific.
Hi, Scott and Dal. We have a request from Gregor here. If you could just zoom in on the sediment one, just so we can sort of suss out what it is.
Will do. I'm sorry, before we leave, can we just get a zoom in onto the sediment to see what the makeup is of the particles? Thank you. They look kind of rounded, so certainly not what I would call pteropod shells. maybe foraminiferal shells. Do you know your sediment?
I do not, so maybe Gregor can chime in here.
Oh yeah, great.
There we go. Okay. Quick.
Well, it looks to me indeed they're all foram shells. No fine sediment. It's all coarse grained. And there are some blackened stuff in there that might be glauconite or something like that is between the grains. But for the most part, these are rounded forams, pelagic and benthic forams. Pretty coarse though.
Gregor, I'm sorry, did you say that they would be largely benthic forams or pelagic forams or some mixture?
I think from the size, it will look more like benthics. They're a bit too coarse, pelagic ones.
Great, thank you. So foraminiferans are single-celled organisms. If you think of an amoeba, if you've ever seen an amoeba in class, forams are kind of like amoeba in that they're a single cell. They can extend the membrane of the cell to engulf organic particles that they feed on, but they're able to secrete a shell or to create a shell by gluing different particles together. So what you were looking at there, basically all this sand or mud that you're seeing are these shells. There's a white structure on the about 3 o'clock on the face of the rock. I said 3 o'clock. I meant 9 o'clock. My bad. You can keep going. Sorry.
Yeah, we see these sediments are all very uniform. That tells us there's really just this single source of sedimentation here.
So we saw one of these before, and there was a question about whether this was a sponge or a possible xenophyphorn. The texture to me looks like it's dead sponge. Chris, can you get a good look at that?
I'm getting a good look at it, but I'm not getting a good idea about it.
Yeah, so it's the second one of these we've seen. We kind of went past it. We'll look for another one. I saw just up ahead, this time on the right, truly the right. I think there was another colony on top of a rock.
Yeah, I don't know. If it's a sponge, I would suggest it would be a demosponge, but because it didn't really look like a hexactinella to me or a glass sponge, but I really don't know.
Okay. This will be good, but what I'm looking at just is going off the top of the screen, but this may be what we were just looking at, so if we're in a position, that's great. Yep, agreed. Well, this kind of looks like a mucus sheet that has been secreted by something and has picked up a lot of organics, or else it was something gelatinous in the water column that has got caught on the rock. But it's not, I don't believe it's the same thing we were just looking at, so we can move on from this. Thank you. I think just to the upper left, there may be another version of what we're, oh, there's a sponge though, right in the center. It looks like a little pogo stick. Now immediately between the lasers. Maybe another sponge went off the lower right. So there's certainly plenty of sponges here. This is also one up off the screen. So there's plenty to look at. No shortage. And Chris Kelly, I'm sure you're going to remember back in 2015, we did a dive where we had lots of these sort of like little bulrush sponges. I forget what they were called. I'm certain that you'll recall.
Yeah, I do recall, but this is Shirley's realm. Shirley, I would default to Piloderma on this one, but maybe you have a better idea.
Yeah, that's as good as a guess as any, Chris. It's definitely it's a demo sponge for sure. I don't remember seeing one on any of the dives that I've participated in.
So there's 2 or 3 of these right in this particular area. Is that a good view, folks?
Yep, really good. Thank you.
Thank you, pilot. I'll point out for everyone listening, that was Chris Kelly at University of Hawaii and Shirley Pomponi at Harbor Branch Oceanographic Institute, the ECC that's out there. Oh, there's a little Galatheid crab scurrying across the bottom. That's a squat lobster. So they were talking about demo sponge, and they're using that terminology to separate this from the glass sponges, which are really common in the deep sea. And I'm certain we'll see some glass sponges at some point. Demo sponge is a group that includes most of the very common sponges that you would find in the world's oceans. Yeah, so yeah, at the top of this rock.
It's got that stalk thing that's up.
Yep, that's where we're going, Chris.
That thing that's up on the upper center. I think that's one of those benthic tunicates, isn't it?
Well, I'm not sure. We're going to try to get a look here. So it's at about 1 o'clock right now, Chris, on this. I couldn't tell if it was one of those, Chris, or if it was actually a dying sponge. Yeah, looks like a sponge to me, Chris.
I don't know, Scott. It's got that opening that looks like that tunicate, though. Yeah, right on the top on the right.
Yeah, I see the opening you mean, but I don't believe that a tunicate would have a fabric quite like this. Of course, tunicates do have all kinds of very interesting colony and body morphologies. They can pack a lot of stuff into their outer wall. A tunicate is it's an organism that is a urochordate. It's very closely related to the chordates, which include all the vertebrates and ourselves. And they can pack all kinds of interesting things into their body wall. So I suppose this is a possibility. Yeah, I guess.
Scott, so
yep.
This is Diva, and when we were working in the Clarion subsystem zone in the Eastern Pacific previously, we'd seen something similar to this, which we had said was from the genus Culeosus. That was totally a guess, because obviously we were working with imagery and hadn't actually collected any. But I would go with tunicate on this one,
okay,
to be very honest.
Okay. Then there should be another opening somewhere on this if it's a tunicate. Now, do you guys think it's alive?
It could be, Scott. It's got a lot of these shells from the foraminifera, I guess, that are embedded in the tissue around it. I think that's my interpretation of what we're seeing. Yeah,
I agree, and that's not an unusual thing for shallow water tunicates to do, to embed various foreign material from their environment into their tissue. I see also that there's some amphipod tubes on the lower side, the darker brown on the left, and you can see some little, what look like antennae or tentacles sticking out. Those look like the heads of some amphipods. Maybe they could be polychaete worms as well. But the bottom line is there are other organisms that are using this tunicate as a host to build their own little houses on. Thank you, pilot. We can move on. And we believe there's a second one of these stock tunicates just up and above in the left corner of the screen. We can move on, pilot.
So we've got some talk in the chat room here about what these rocks are still. I'm continuing to see a lot of pillow lavas. I think many of us are beginning to believe that these are in fact intact lava flows that would have come out when a water flow erupts underwater. The outer crust tends to cool, and then it'll burst through and make these pillow structures. Of course, the black stuff on top here, this rine, is our iron manganese crust, and these crusts can be up to a few tens of meters thick in some parts of the world. Again, they tend to form on the slopes of these volcanoes, and really they need a bare rock surface, like we would expect when we have currents pushing the sediments out.
Alan here, can we maybe see about grabbing some of these looser boulders there that the lasers are pointed at right now? They seem to be, they seem like they might come loose.
Yeah, pilot, can you try one of these loose rocks here?
The one at 3 o'clock looks nice because it seems to be a bit blacker than some of the other ones. I'm not sure if that's because of less sediment cover or.
Yeah, what about the one over around 3 o'clock?
Yeah.
Yeah.
That moved. This one.
Alan, what do you think?
The one that's just to the right of the lasers now.
Yeah,
That one.
Yeah.
Yeah, I think that's it's worth a try. Let's take a look at it and pop it in the basket.
Great. Either way. Ready? Great. Oh, looks good. If we can get it in the box. Great.
Yeah, that's a nice sample. Definitely a manganese crust on it, but hopefully not too much that there won't be some usable rock in there.
Great. Yeah, so as Alan was mentioning, these rocks all have this manganese crust on it. We'd hope to get into some of the basalt inside to look at the geochemistry, to look at the geochemistry of these rocks versus those in the Anshan Java Plateau, and also the rest of the Manihiki Plateau, and hopefully get an age date as well.
Yeah, and given the morphology of the northern plateau in this region, it seems to be far more plateau shaped as opposed to some of those seamount structures and the pancake volcanoes and so on that we're seeing on the multi-beam. So I'm hopeful that we're looking at plateau-aged volcanics here, but it is on the edge of this rift system also. So it is possible that it's later stage volcanism associated with the rifting. In any event, the results will be very interesting.
That's great. Thanks, Alan.
All right, so our first collection of this Mountains in the Deep expedition.
It was a rock.
It was a rock. I'll bet there was some biology on it. Very small.
Dare to dream.
Dare to dream, exactly. We are at 2,483 meters, and we are diving on a site we have called Tuku. We're at the northern edge of the Manihiki Plateau, and we're just north of the Cook Islands, EEZ, and the Marimoana Park, which is a equivalent, I guess, to our monument, I believe. It's a protected area. Copy, pilot, thank you. Is this the light brown object going for? I see, yeah. So to the left there is another one of those tunicates. Oh, great. This is what Chris Kelly was referring to earlier that he thought he'd seen on a rock. So this would be a xenophyophore. And I was referring to earlier, or I should make sure that nobody is contradicting me and everybody agrees with me, but my impression of this is that it is xenophyophore. So this is one of these single-celled organisms that I was referring to earlier, and it's quite surprising given its size. I don't see the lasers right now, but I think this is probably a few centimeters across. There you go. So those centimeters are 10 centimeters apart. Excuse me, those lasers, those two red dots are 10 centimeters apart. And so that's probably 4, maybe 5 centimeters at its widest dimension. You can see it's growing up in that sort of a plate-like format. So this is a cell that's basically trapping various other shells and particles and gluing them together. And it basically feeds on organic material that is small enough that it can take into its cell membrane and digest. So wonderful. That is an erect xenophyophore. And there was some question earlier, I forget who had posed it, asking can xenophyophores grow on rock? Don't they only grow in the sediment? And here you can see that indeed this is growing from the rock. You can see there's all kinds of other little stuff that's growing in the rock. It's just amazing how much biological diversity is down here. A lot of it is related to foraminifers and single-celled organisms. There's some very small sponges and worms and crustaceans. A lot of stuff that we don't get to see when we're passing over a little bit more quickly. Thank you, Pilot. We can move on. No, that's the same tunicate that we were just saw earlier. Oh, but here's something interesting. I think this might be another little tiny carnivorous sponge. It's really small. You just kind of see it waving back and forth there. It's branching, so it's different from the one that we saw earlier, but it's really small. Just kind of a quick impression, Del. There's a lot of stuff, a lot of biology that's using all of this sedimented particles for their own needs. Yeah, that's. I think that's going to be so small we're not going to get much out of it. Thank you. Okay. Yep.
So Scott, are they deriving nutrients then from the sediment? It seems like mostly they're attached to the bare substrate, of course.
Right. Yeah. So for the most part, I think what they're doing is using it to construct some kind of domicile. You know, it's their little house. They build tubes and so on. However, any organics, bacteria that may be associated with those shells certainly is fair game for organisms that will ingest the sediment. So if we see some sea cucumbers later on, for example, that's kind of a typical mode of feeding, is just to ingest wholeheartedly all the sediment that's there. Whatever organic material, nutrients are mixed in with it or on it, they will digest.
Okay.
And you can see in some places here the coloration of the sediment's a little bit different. It's a bit darker in some places and lighter in other places. I don't know about here, but in other places like in the North Atlantic, that's evidence of some fresh deposits of phytoplankton. So we have a couple of biology items here on the rock, and it looks like one of them may be a Brazingid sea star, that long arm pink thing. We'll talk more about that in a second. And then maybe in front of it is our 4th or 5th stock tunicate. Please do the oh, you see the sea cucumber. I'm sorry, I'm looking up. Yeah, absolutely. Yeah. Good call, good call. So right in the center of the screen, just to now between the lasers. What perfect timing, wasn't I just saying?
It was quite a transition there.
Yeah. So this looks like it's going to be a sea cucumber once we zoom in. And instead of me describing it, I think Diva knows a lot more about sea cucumbers than I do. Or am I putting you on the spot, Diva? I'm not hearing Diva, so let me just say while we're looking at this in close-up, so this is actually related to the sea star.
Sorry, I'm just trying to look it up. Keep talking. I'll get back to you.
Okay. So this is an Echinoderm. It's related to the sea star, believe it or not. It seems a lot more fleshy, and it is, but all Echinoderms are classified together. Sorry, my eyes just noted that there's 2 octocoral polyps right next to it, so back to the sea cucumber. they have small, what are called ossicles, hard parts embedded in the skin, and so they have this spiny skin. And this one, you can see its tube feet are modified into what look like legs. It's almost like legs of a centipede or a caterpillar or something. And at the anterior end, which is at the top, it's got that hood, and the mouth is on the underside of that. And so this is one that would just be ingesting chunks of sediment and digesting the nutrients out of it. There's a lot more to talk about, but just because I see that there's a coral polyp here, I just want to say something about that before we move on, and
Hey,
I know
Scott,
we'll
wait,
see
so
more.
quickly, before you talk about the coral,
Yep.
I think it's from the family Latemagonidae. We've seen similar ones in the Clarion Toxin Zone, but not quite identical. But that's the best I can go, family level.
Yeah, this is not a morphology I recognize. There's a lot more of these podia, these leg-like structures extending from it. Interesting close-up. It almost looks like there's more very small tube feet on the modified tube feet. Seeing all these little white, almost look like little fingers sticking off the back.
Yes, Steve, a few weeks ago, this is Chris. A few weeks ago, I was speaking with Dave Paulson at the Smithsonian about holothuroidians, and that observation about the large number of tube feet is important, and I think that supports Diva's identification that this is a potential Latemagoniid.
And there's something else underneath it. You can see, I think it's a polychaete. Just on the left side towards the top, there's something creamy white just sticking out of the edge of underneath that holothurian, and I suspect maybe there's a polychaete under there. And I suppose while we got this nice close-up, you can see through the tissue of the holothurian these kind of whitish coiled tubes. That's the intestine, and so that's the sediments that is ingested being processed in the intestine. And so this thing just sort of continuously moves its way through the environment, chomping on the sediments. And, you know, as a geologist, I'm sure you know about reworking of the sediments.
Sure.
This is one of the organisms.
Bioturbation in action here.
Exactly. So very nice. The general term for this kind of mode of feeding is deposit feeding. And I was just going to briefly say, we'll talk about this more as we go through the dive, but there are some holothurians that are known to be selective deposit feeders. Selective deposit feeders. So they're not just going to munch on any old sediment, but they will preferentially take the sediment that has the freshest deposits of phytoplankton falling from the surface.
They do a little taste testing.
Exactly. They are capable of doing taste testing. So what's really interesting about this coral that I was pointing out is that it's only 2 polyps, and this suggests it's a very recent recruit. So in the reproductive cycle, the life cycle of corals, they are sexual organisms, so they produce sperm and eggs, recombine the sperm and eggs, and you get an egg that develops into a larva, and that larva can move through the environment. Some of them swim, some of them crawl. When they find a spot that seems to be suitable for life, they then metamorphose and start growing into these colonies. And this one is a colony of only 2 polyps so far, but it will continue to grow and bud new colonies and get bigger. So interesting observation. It's too small for me to really say what kind of octocoral this is. Some of the possibilities include an Anthomastus, the genus Anthomastus, or even something in the Paragorgiidae, but I'll look to see if our scientists on shore have any sense. Tina Molodtsova is saying something dimorphic, and what she means by that is that there are 2 kinds of polyps. So what you're seeing here are the main feeding polyps, use those tentacles to grab food and stick it into the mouth. But then there's another type of polyp. It's really small, has no tentacles, and it really looks like kind of warts on the surface. Those are called Siphonazoids, and they can be used to funnel water into the colony or to store gametes and eggs and so on. Thank you for that view. We're good. And we were on our way to what I believe is a Brazing asteroid. That means it's a starfish in the group, either a family Brazingidae or a larger group, Brazingia. And we often hear from Chris Ma at the Smithsonian to educate us some on these sea stars. And if Chris isn't available, then maybe Chris Kelly can tell us something about it. Malcolm Clark from New Zealand has said that 2 polyp coral looked like an Anthomastus to them. Thank you, Malcolm.
So Chris said he was having some trouble connecting there, and I guess I'll just confirm for him, I guess, your ID, Scott. So this does look like a Brazingid. One of the things, the way you can tell the Brazingids from the fraility, which is the deeper kind of Brazingid looking like sea star, is the lack of ornamentation on the arms. So this particular one has these sort of ribs, if you look, that are going across the arm. And if I understood Chris Maw's lessons that he was giving me, then that would identify this as in the family Brazingidae. But I don't think we can go further than that. There's a number of genera that are very close in appearance, and so I think that's all the further Chris would go to.
Thank you, Chris. So I'll note a couple of other things on the rock here. Just that went off to the left of the screen, there's another one of those really tiny carnivorous sponges like we were trying to see before, really small. And then these 2 brownish globular things, these are 2 more tunicates. As we were looking over the surface of this Brazingid sea star, I was able to look in the opening of one, and I clearly saw the pharyngeal basket. Basically, these tunicates are suspension feeders. They can generate a current that's drawn in through the opening that we were looking on that stocked one, and then they have their pharynx basically has a whole lot of holes in it, and it's coated with mucus. And so as water comes in, any particles get bound up in that mucus, and they can ingest it. So I definitely saw that in these 2. One last thing about this Brazingid sea star. So earlier in the dive, we saw a crinoid, and it looked kind of the same, had lots of arms, and they were up in the water. And I said that you could see in the center of the arms was the mouth. Well, this is a more typical sea star. The mouth is oriented down toward the substrate, so you don't actually see it. It's against the rock. And one thing I know Chris Maas taught me from listening to other dives, you see all these little spines that are coming off the arm. You see they're kind of smooth, and that's because there's actually tissue on those spines. And he describes them, or he has said from another paper that he's read, that they're kind of like socks on the spine, but the socks are full of Velcro hooks. So each of the little hooks on each of these spines can capture prey from the water column, and then they're transferred to the tube feet, which are underneath these arms, to the mouth, which is against the rock. Thank you. Very often you see these Brazingids, they'll have their arms right up off the rock, so they look a lot like crinoids from a distance. And there's another one of those little bulrush sponges waving in the foreground. There's also a xenophyophore next to it, a stalked tunicate, and I see a brittle star wrapped around that stalked tunicate. So there's all kinds of stuff down here. Thank you, pilot. Les Watling has made an observation in the chat room that gets to the slow rate of life in the deep sea, and he says he wonders how many months or years before that Brazingid sea star moves its arm off of the tunicate siphon opening. Things don't happen really quickly down here. I've already noted how cold it is. Again, the temperature is 1.79 degrees Celsius, so it's really cold. And what that typically means is that the metabolic rate of the animals is depressed. They don't need as much food.
Right.
Good thing. There's not very much food, so they're adapted to that. And one of the ways they're adapted is they're not highly energetic, so they don't move more than they need to.
I guess they're not chasing prey, and prey isn't chasing them. So.
Not for the most part, although you do see a few predators down here, certainly among the fish.
Right.
Some of the worms and the crustaceans. I think a lot of them are sit-and-wait predators. So this looks like something a little bit different, very fuzzily branched brown structure. I have a sense of what it is. Get a little bit closer. If anybody on the shore wants to identify this before me, please feel free.
Yeah, what's that moving there on the right
Stumbling
side?
snail on the right.
Yep, okay, thank you. So we'll come back to the sessile thing that doesn't move, but you can see that there's something that seems to be bouncing around here on the bottom. This is actually a snail, and that long sort of wormy-like thing you see is its foot. So the foot of a snail, when they're a little bit more calm, is what they're sort of moving over the substrate with, but it's a very muscular foot, and so it can use it to sort of push itself off against the bottom and trying to get away. There we go.
Wow.
Nice. That is terrific. I believe I even see eye spots at the base of where the antenna would be. There's 2 black dots. And we've seen this snail a few times now, and I forget the name. I wonder if anybody on shore can remember the name.
Yeah, Scott, it's in the family Margaritidae, believe it or not, and the genus is Gaza. Mary Wickstein just published a paper on the ones that were observed off of Hawaii, and I can't remember whether she went to Gaza Daedala, but definitely in the genus Gaza, or at least it certainly looks like it, because the appearance is identical to the Hawaiian one.
Thank you, Chris. So the idea is probably that that's an escape mechanism that is being disturbed, and so it's certainly a much more quick way of travel than your typical snail. The other thing that we're seeing here is a black coral. So this is our first black coral to dive. On dive number 1, we saw, in fact, black corals were the dominant kind of coral we saw. Most of them were wire-like whips. We saw one that was quite yellow, and now we're seeing one that's brown. So we use the term black coral. That's the general descriptor for the entire group. It's an order of cnidarians because the skeleton is black. Sometimes they're also called thorny corals because the skeletons are also very spiny, and they use the pattern of spines, the size of the spines, and the shape of the spines on the skeleton, plus how often the skeleton branches and some other details to identify the genera and the species. But what you can see is that the tissue, the flesh that is overlying that skeleton, comes in a lot of different colors. So we've now seen them in white, yellow, and brown. There are also oranges and reds and so on. No, this is pretty good. So we've got to zoom in here, and so much like the octocoral I described earlier, this is a colonial organism, so you have a whole series of individuals that are all connected to each other, and each individual has tentacles. Now, these are, in the hexacoralia, they have 6 tentacles, 6 tentacles surrounding a mouth. And you can see every once in a while, you see a little sort of raised pinkish white dot. That would be where the mouth is. You can see that on these main inner branches, it looks like these polyps are really crowded close together, and they're a little bit more spaced on the branches. One of the things that we would be doing in a close-up like this, in addition to just trying to get some general identification information, is to see if there are any other organisms living on it, like worms or shrimp or crabs, and I don't see anything right now. Thank you. Tina Molitsova, I mentioned her earlier saying she was an expert in tube anemone. She's also an expert in the black corals. So there she goes. So she's given us an identification of the genus Trisopathes. So this is the genus Trisopathes for this black coral. And that is great. And I'm not exactly sure why it is that on the older parts of the colony, which would be the main branches and the main stem, the polyps are so apparently jammed together. That might be because there is budding of new polyps between older ones, perhaps. Now, I think it's just that, yeah, the polyps are so thick it looks like there's something on there and there's not. So we've seen some interesting biology here, so I think it's probably a good idea for us to get a sense of how much bottom time we have today, so everybody is well prepared for how much distance we might be able to cover versus the close-ups and collections. This is something different as well, this purple thing right in the center. And, okay, sounds good. Looks like we'll be talking about a bat star or something similar, a slime star in the future. So I guess I should wait for the next nav to come on before asking this question. Copy that. Nav's watch lead, if you could give me an estimation of time off bottom so that I could relay that to shore, please. Four hours, thank you. So shoreside scientists, we have about 4 hours of bottom time to go, so just for your various planning.
Stand by, guys. I want to confirm that. Thank you, Fernando.
So apologies to get you all excited. We have 2 hours left in the dive, 2 hours to off bottom. It's that was first estimate was probably to recovery time on deck.
Right.
Okay, so we have
I think the East Coasties just breathed a sigh of relief.
We absolutely did over here.
Yeah, so as we said at the beginning, we had a very long transit to get out here into this remote part of the Pacific, and so we started a little bit later today. So we're looking at a sea star here, but if we just back up, is that a tiny little crinoid? I think there's a tiny little crinoid just to the lower left of, it's against the sea star. Yeah. And I see some xenophyphores also coming off the rock here. Yeah, so I mean, if we just look at the sea star.
Sorry, I'll be Chris Ma again. This is a hymenaster. It's in the family Pterasterid, or as you said, a slime star or a cushion star, that whole group. And I think you'd already pegged it as a slime star well before they even got close to it. But anyway, this has got a very, very gelatinous texture. They tend to have very short, stubby arms like this one does, almost transparent. And then that big, huge opening on the top, and you may see it actually open and close as it's respirating. But yeah, hymenaster, some species.
Thank you, Chris Kelly. And we see just in front of it, the lower left, there's a tiny little crinoid there, and it may be a stalked or it may be a non-stalked crinoid. The crinoids, they're called sea lilies or feather stars. The sea lilies are the stalked ones. The feather stars don't have a stalk, but they begin life with a stalk, and then they would separate from the stalk and take up a free existence after that. Thank you. And then just above this sea star, there's another fleshy pink organism that is likely a holothurian. And I also saw a little crab underneath the rock where that holothurian is. So this looks like a different sea cucumber than we were looking at before. So the earlier one, you remember, had all of those different leg-like structures arising from it called podia. And this one is much smoother, but it is still a sea cucumber. It is still a type of Echinoderm, deposit feeder. Diva, do you feel comfortable identifying this one?
No, I do not.
Yes. Well, we've got some...
Be brave, Diva, be brave.
I think we have some good images, so let's just nip over to the right here and have a look at that, what looked to me like a squat lobster. And you can see the lasers on there, so that sea cucumber is less than 10 centimeters length.
I did not yet.
Oh, thank you. Malcolm, I see your comment, and I copy that. And so this looks like a munid squat lobster to me, which are a pretty typical group of squat lobsters that live on the bottom in the deep sea. So we're good when video is good. So we're at 2,470 meters depth, and we haven't gone very far since we settled down, and so we're going to see if we can try to make some hay and get up the side of this ridge. There's a lot of interest in seeing what happens as we change depth zones and maybe get up to the top of this feature.
Yeah, the geology has been remarkably uniform so far. Doesn't look like there's a lot of kind of three-dimensional structure here. There are a few places for that squat lobster to hide, but not compared to some other sites that we'll probably see.
I see another squat lobster on the rock to the left. It looks like the same that we just imaged. And we can keep moving, pilot. I'm just noting it. There he goes, turned around.
So this is Deb and Gregor over here. We're just looking at the geology right now, and we're noticing there's a lot less sediment than there was. It looks like there's a little bit more rubble than there was before. Del or Alan, do you guys have any ideas on what might be changing?
Yeah, I agree, Deb. It does look like this little patch here, at least, is a little more broken up than what we were seeing down slope. Of course, as you go up the slope of a volcano, sometimes you'll see these little slope breaks. It's a little bit difficult to see where we are, but we might just see a sort of transition back and forth. That would be my guess. If you're watching from home, you can always go over to camera 2, of course, and see the view from Sirius up above. I find that useful as a geologist sometimes to get a sense of the terrain. Sometimes when we're focused in here on D2, it's a little hard to get a sense of where you are on the slope.
That was a little amphipod that we just saw swum by the front of the camera. We saw earlier 2 amphipod crustaceans on the stalk of a carnivorous sponge, and so that would have been the same group, not necessarily the same species. Amphipods are very common in the ocean and in the deep sea.
Yeah, this is Alan here. I noticed as we were coming up, there were a couple of ridge-like, almost ridge-like features that we passed over, and on the edge of this escarpment, we're in quite a heavily faulted part of the Manihiki Plateau. So I think it would not be uncommon to have these tilted fault blocks. It's extensional faulting, so kind of the Manihiki Plateau was being pulled apart, and so in the intervening dips of the structural highs, I think is where you would get sediment filling in more naturally, and then you might have some more structural highs on either side of these smaller ridges.
Well, it may be reading too much into it, but to go along with this theme of is there increased rubble here, there seem to be fewer of the sessile fauna here. I mean, we weren't seeing a lot, but I'm not seeing any right now.
We'll see if this continues and if we come into something different here as we keep going up slope.
Thanks so much. That was really, really helpful.
So, Alan, would you, for a second rock sample, would you prefer we wait till closer to the end of the dive, or do you have a preference?
Some of the ones that, when the camera panned to the right, it seemed to be there seemed to be some kind of fairly large angular blocks that might be good there. How many rock samples are in and around the size we've taken can the ROV handle? Haven't been briefed on that detail.
We've got 2 rock boxes, and if I'm really nice to Scott, we can get a small rock, maybe in a bio box.
So far, he's been pretty nice.
Oh, I guess the real question is, would you like to try to get up slope a little bit more, and then look around, or?
Yeah, I wouldn't mind picking one up here, and then as we move up slope, I'd like to see if we start to see some more coherent fall planes, which is something I would expect here. But they may not be here, so maybe picking one up here, where they are. Yeah, you can see up in the top right of the screen there. There's some kind of nice-looking larger blocks. And so maybe picking up one of those might be a good option. And then if we get up and see something on an escarpment of some sort, we could maybe grab one of those if possible as well.
So, pilot, this is watch lead. We're still looking for a rock. So some of these loose, angular rocks, the bigger the better that we see up here. If you can see if one of those is available, that would be great. Yeah, that pile would be fine. The bigger the better. Yeah. Yeah, Casey's just reminding us, Alan, that we get a third rock. It'll have to be smaller to fit in the bio box. So,
Understood. Let's hope the good one isn't up at the top. We might have to throw one of the earlier ones out.
Well, the alternative is you take a small one now, but then we have to definitely use a bio box for it if you think there might be a bigger one up above.
Well, maybe we should save it on. I mean, they look quite similar to what we've collected. So maybe as we go further up, we save the larger sample for further upslope in the event that there is a better looking sample further upslope.
Okay, roger that. That's correct, pilot.
Well, looks like maybe another bamboo coral, although always dangerous calling something out so far in advance. But I see an erect whip-like structure. So it's either a coral or a sponge. But it looks to me like there are polyps extending off it. And the fact that the main stem isn't quite perfectly straight suggests bamboo coral to me. Can we get a zoom on that, pilot, please? Thank you. So earlier in the dive, we saw a branched bamboo coral. It had two branches, and the lower part of it was, the tissue was partly stripped off, and there was a brittle star there. This is another bamboo coral, and you can see it has some prominent spines. Again, the flesh is transparent, so you can see through it, and you can just see that white axial skeleton inside of it. This, I think, is different from what we saw earlier. And what I am looking at, I'm trying to see evidence for the sclerites, those spine-like structures in the polyp body, so the part that actually attaches to the main stem. And I saw some curved ones, kind of oblique in the earlier one. I don't see that now. So that's good. We can zoom out when you're ready so that we can make good transit time on our short dive. as a very small holdfast. It really doesn't extend over much of the rock. The holdfast is the base of the colony that is glued to the rock. And another thing I note is that there don't seem to be large polyps on the lower quarter, let's say, of that colony. So there's some reorganization going on in the taxonomy of bamboo corals, but formally as of right now, that would be something that would be put in the genus Lepidopsis because basically it's unbranched. There are a couple of other details that I'm not going into, but it's an unbranched member of the subfamily Keratoicidnae, so we would call that Lepidopsis, even though we already know, based on genetic studies and morphological studies, that there are a lot of different ways that bamboo corals can be unbranched and be not related to each other. So here may be a second individual of that same species, and I see another one of the larger pink holothurians, sea cucumbers, excuse me, just in the center. So yep, that's in this. You can also see in this bamboo coral it's unbranched, and once again the lower quarter of it has no polyps. So I'm quite confident that's the same thing. And here we have one of these sea cucumbers that does not have a lot of the foot-like podia extending from it. Now I'm not certain. Have we seen this one today, or is this now a third or fourth type? Yeah, I think this is a new one for the day, unless it's just a much bigger version of that one we saw earlier that was less than 10 centimeters. I suppose that's possible. What do you think, Harbor Branch? Have we seen this already today, or is this a new one?
We think it's a new one that we haven't seen today, but again, I wouldn't be able to definitively say what genus this is, but I guess Malcolm's guess, which was Bathyplodes he put in earlier, was a pretty good guess, I think, but I think this is different.
Thank you. Thank you, Pilot.
Yeah, we agree with Ziva that it looks different from the previous one.
So that's good. So we've got 3 or 4 species of sea cucumbers already in this dive in the first, what are we, an hour in or so? So again, there's this question of how much of this is relatively loose rubble versus cemented in place. And the more loose it is, the harder it would be for anything attached like a coral or a sponge to grow without being dislodged. The more cemented it is, you'd expect that they could make a living here as long as there was enough food floating by in the water column.
Yeah, this rubbly appearance is certainly continuing that we've been seeing now for a while.
This may be coming out right up in the center. There's something else on a stalk, and it's either a crinoid or a crinoid on a dead stalk. Chris Kelly, if you want to continue in the Chris Ma role and tell us something about this animal. There may also be an urchin.
No, I think I'm going to be Charles or Chuck Messing right now. He's the crinoid guy. And basically my guess is that this could be Naumachocrinus. No, maybe not. Sorry, maybe a Hyocrinus would be my guess. Yellow, I can't tell if it has 5 arms, but it should. And not much of a calyx right at the base of the arms. So that's my best guess. Or the other possibility is a bathycrinid. Maybe bathycrinid's better. Sorry.
So Chris is at least confirming for us that it is a crinoid. So this is another kind of echinoderm. We're having a little invertebrate lesson in the phylum Echinodermata. So we've seen now sea stars, we saw a brittle star, we've seen a holothurian, and this is a crinoid. What am I missing? We're only missing an urchin. Oh, look, just off here to the left, I think I see urchin spines, which means we have all 5 classes of echinoderms represented on our dive so far, so that's cool. But one thing I want to, can we zoom any closer onto the stalk of this crinoid? You see there's 2 little tube-like things hanging down. I believe that those are specialized predatory gastropods, snails. And there's a whole family of snails that are specialized to feed on echinoderms, and they stick their proboscis basically into the crinoid. I think that's what we're looking at there. There's 2 of them, and I saw a few of them further down on the stalk. And then there's a whole bunch of, I believe these are hydroids, a kind of cnidarian related to jellyfish and corals that are building their little tubes on the stalk of this crinoid. So we saw a crinoid earlier, and I was comparing it to the Brazing Asteroid, the sea star. So the crinoids are echinoderms that have their mouth facing away from the bottom. So the mouth is in the center of the arms there, and you can see the very fine branches. Basically, those are the tube feet. So if you've ever picked up a sea star and you see those tube feet under the arms, it's moving along substrate. Well, those are the tube feet of the crinoid. That's a good clue to you that they are all related and they're echinoderms. So if we could get a look at that thing on the left, I think those are spines of an urchin. And I'll go back to Chris Kelly because he knows a lot more about the urchins in this area than I do.
I was going to comment on the crinoids. Now that I can see the arms a lot better, I'm pretty certain that this is Bathycrinus, or a bathycrinid at least. And interestingly enough, the ones we often see have got those hydrozoans growing on the stalks. Now this guy could be a Spitodiadema, I think, or Cenopodyna, but the really long spines on there, I got to look this one up. Sorry, Scott.
Oh, that's okay. We can't really see it. It's tucked in behind a rock, but I'm assuming that's a sea urchin with some very long spines. And I'm also now looking at the sort of golden brown tube-like thing just in the foreground on the left, and I can't tell you what it is. Something agglutinated, and I see there's either tentacles or spines coming out the top. So plenty of mysteries down here, lots of diversity. Certainly all the echinoderms are represented, so we can move on. Thank you.
So if you're just joining us here, we're diving today in the Manihiki Plateau on the Tuku site. We're diving on the eastern edge of a large plateau-like feature here. The rocks you're looking at are all manganese-crusted basalts. And we've been seeing various texture to them. In some places it looks like they're intact flows. And here we're a little bit more into an area where it looks like there's still quite a bit of rubble. And most of the sediment we think is probably forams that have been deposited.
So Malcolm Clark, who's at the NIWA, the New Zealand Institute of Water and Atmosphere, I wonder if you can tell us what your impressions are so far of this dive. You've worked obviously down in this part of the world a lot more than we have. Is this surprising, unexpected, what you expected, or you didn't know what to expect?
Yeah, hi everybody. Yeah, so far because we didn't really know what to expect, it's been very interesting, but not perhaps unexpected in that there's not much variation in the topography, so it's a fairly uniform substrates down there with the broken up crust. So we're not seeing a lot of relief. There aren't many cavities or ridges or other features which would normally be where we'd expect to find more of the larger aggregations of sponges and corals. So it's pretty much what we get further south in the Pacific as well, given this type of substrate. But I must say we're very, very impressed here watching the high resolution camera feed that is really able to give us a good idea of what some of the smaller animals are looking like.
Thank you, Malcolm. We're looking at another crinoid here. I'm pretty sure that this is different from the previous stock crinoid we just saw. This one does have 5 arms that are then subdivided, and I see there's an amphipod just underneath the calyx. Chris referred to the calyx before. It's that thickened part where all the arms join, where basically the very small stomach is housed.
Yeah, so Scott, I'm pretty sure that this is the Hyocrinidae with the 5 arms, the yellow color, and the similarity to similar ones that we documented elsewhere. This does not have those disc-like rings right underneath the calyx where the arms connect. And so that's going to mean that it's not that new genus that just got described, the Tiburonocrinus. So this is just Hyocrinidae for now.
Thanks, Chris. And yeah, I know what you mean. Those rings are kind of like necklaces that would be right where that amphipod is sitting, that pink crustacean. Really nice view, and again, you can see the small little tube feet extending from the arms and the pinnules, which are the side branches on the arms. Terrific. We can move on. Thank you. Astrid Leitner from University of Hawaii noted that as we were zooming in, there was a squat lobster on the rock just behind there. I can see it's against one of the really dark faces. It's pink in color. And we can keep going up slope, pilot.
May I put in a 210 This is Chuck Messing.
Somebody on shore, I think, has their microphone unmuted.
Should be. Let me check.
I think that...
It's not muted.
We're passing over another one of those flat xenophyophores, the foraminifera that form the sort of plates. And I see another one of the crinoids, the first crinoids that had all the hydroids on it. But just behind, right at the lasers right now, I think might be a sponge that we haven't seen, or maybe it's another one of the tunicates that we were looking at earlier. So if we get just a quick zoom on that, please. Okay, now this time, I'm sure it's a sponge, Chris.
You're right, Scott.
Thank you.
So this is a glass sponge. It's one of the stocked glass sponges in the family Euplectellidae, and I'm sorry, Rossellidae, and it's in the genus Calophacus. The very smooth surface on the ball there suggests that this is Calophacus subgenus Oxydiscus, I think, but I'm not positive. I got to look it up. Anyway, definitely glass sponge, family Euplectellidae. Family Rossellidae, genus Calophacus.
Thank you, Chris. And that might be an isopod that's crawling around on the back of it. And before I say anything more, I see that Deb Glickson from Harbor Branch wondered if Chuck Messing was trying to call in. So Chuck Messing is at Nova Southeastern University in Florida, and he's an expert on the crinoids. So if he was trying to call in, we'd love to hear from him about some of these observations that crinoids are making. And there's a, just on the right. Yeah, go ahead. Go ahead.
This is Chuck Messing.
Hi, Chuck.
How much of a time delay there is. Hi. You're right about that Hyocrinidae, and I had a friend's birthday celebration, so I just had logged in and caught a glimpse of the crinoid before, which looked like Bathycrinus with the very small conical calyx and the 10 arms.
Great, Chuck. Did you? I'm not sure if you saw. Maybe it's this one you were talking about, the one that had the gastropods on it. Do you know anything about those, or can you share a little bit more natural history?
Right, that was the one. Interesting that it was attached to a hard bottom, so because I only got a quick glimpse, because Bacchus is often rooted in the sediment as well. So that was my first guess on such a short glimpse, but right on the second one with the 5 undivided arms, the very few finules, that was definitely a higher Crinoid.
Great, thanks, Chuck. Yeah, please continue to call in as we see stuff.
Very good. I'm going to be in and out, but I'll try to stay on later tonight.
Thanks, Chuck. So I was Chuck Messing at Nova Southeastern University, who's a world expert in crinoids and deep sea biology. Done a lot of work in the Caribbean. So yeah, I'm sorry, I noticed something else. I was going to say to you, this is a really big rock now.
Couple bigger rocks here, yeah.
I hope that's not what Alan was hoping to see.
It will not fit in the rock box,
Okay.
Alan. So.
Just the one so far. So we've just passed 2,450 meters depth. Temperature is about 1.8 degrees Celsius. We are diving on a site named Tuku, and we are just north of the Manihiki Basin and the Maria Moana Park of the Cook Islands.
So we're coming up to a little bit more of a ridge here.
Yeah.
We've seen a few of these small ridges already.
So this other stuff that's rubbly-like, would that be a collapsed ridge or breakage from the ridge?
Could have just broken off the ridge, although stuff we've been going through for a while has been completely covered, so I would assume a lot of that is coming from further upslope.
I'm seeing another one of the large Brazingid sea stars that we imaged earlier. So that's the thing in the background. In fact, yep. So Caitlin's pointing out to me that there's a second one up a little bit further, and then I'm seeing several of these stalked Tunicates. They look like little goblets on a stalk. Actually, are these the sponges? So this is the problem. The earlier one I saw, I thought was the sponge that Chris was just recently describing. So now I have to be careful. See if these are sponges or tunicates. It's interesting that they have a very, very similar morphology.
Yeah, when you look on Sirius at this view, you get the sense this side of this cliff here is sort of broken away. What we saw as a ridge, you get a more of a scaled out view of it.
So this is nice. This is
Alan's here.
Go ahead, Alan.
Yeah, just again, looking at the overhead view there, you can see there's a good continuity of linear features through the rock there. So I think we're looking at structural features within the rock associated with some of the faulting that we see from Manihiki, this part of the Manihiki Plateau. If possible, I'd like to get a sample. Maybe you can see there's some angular pieces at the base, right where that. Almost dead in front of the ROV now, right where the kind of fault linear feature. You can see there's sediment is infilling in the kind of linear feature, and I think that stuff that will have come off the fault plane. So if at all possible, a sample up along the base of the escarpment there would be a little further to the left of where we are now. Where we're facing now would be very nice.
Okay, thanks, Alan. So, yeah, pilot, we'd like to try to get a sample up the slope here closer to the base, if we could, a rock sample. And again, since we've got a rock box open, the bigger the piece you can still get it in, the better. Yeah, I think the bigger one there. Yep.
You got it?
So. Port diamond. How does this look, Alan?
Yeah, I think.
Is there a bigger one that we could still grab? I think that might be preferable. We're expecting these will be.
Yeah, there's a more angular-looking one immediately to the left of the laser dots there.
Right there, Alan. Yeah.
Yeah, that's the one. If it's that it's pointing to there. That
All
one
right.
would be.
Looks like it is loose. Great.
I don't think you'll ever see a biologist put their sample back down for another one. Once you got it.
Oh.
The rocks don't mind. They don't feel the pain.
Oh, that's right.
That is a rubble pile, isn't it?
Yeah, if you've been watching some of the other cruises before ours out here, often they've had trouble grabbing rocks in these manganese-crusted areas. Here we've had lots of rubble, and yeah, as Alan's pointed out, perhaps there's some structural control here that's causing some faulting, giving us these rubble piles. Oh, that one's breaking apart.
Well, we know we don't want that one.
Okay, so let's see if we can find one that's a little bit more solid here, I guess. Sorry.
It's quite amazing that it's that friable. It's
yeah,
it be quite heavily altered.
I was surprised as well. I thought it would be a crust on top, but it really crumbled onto that claw. Yeah, this one looks good if we can give it a try, pilot. Thanks.
Yeah, I agree. That one looks a lot more solid. Yeah, looks good to me.
Okay, great. All right, it's in.
Thank you. That's great.
Thanks, pilot.
So we have 2 rock samples today.
You should probably just get all rocks today. What do you think, Scott?
Well, there's a lot of rocks to be had. In all seriousness, that's great. The geology was a high priority. It's one of the reasons we chose the Pacific dive track that we have today. Take dive site. And you've heard a lot of information from Alan Murray about, Murphy, sorry, Alan, about the geological setting up here in the Manahiki Plateau, and the interest in getting some ages and other geochemical information. These rock samples will be really useful, and they will go back to Oregon, I believe,
right.
Oregon State, and be available for different scientists to request and do studies on. So we're headed back up slope. We're at about 2,446 meters depth. And we have just over an hour in bottom time. So let's see how much ground we can cover. Oh, yeah, good call. Yes, very nice. So we're looking at another kind of octocoral here. This is in the group, the Panatulacea, which are the sea pens. And this one is really interesting because it's a relatively recent discovery of a group that are referred to as the rock pens. Now, in general, you're all taught in your invertebrate zoology classes that sea pens are specially adapted octocorals, and I have to point out that there's also a worm on the back of this thing. That's what that lump is on the left side. It's a little polychaete worm. So sea pens are specially adapted octocorals are adapted to live in soft sediment. So the base of the colony, instead of the holdfast, I was describing a holdfast before as the part of the skeleton that's glued to a rock. Instead, their base, they have a muscular inflatable structure called a peduncle that normally anchors them in the mud. But this group, the rock pens, they're much more like plungers, and they kind of act like a little suction to kind of hold it to the rock. And you can see it's got one axial skeleton running up that center part of the colony. And sea pens are also interesting because there's one giant polyp, the giant polyp being the one that has that inflatable base, and then these other polyps are growing off of it. So it has a different kind of a growth structure than most of the other octocorals. Thank you, that's really nice imagery. We'll be able to get good identification from that. I suspect this is a genus, something like Cophobelemnon, or Calibelemnon, excuse me, Calibelemnon. Good spot there, pilot. I did not see that. And here's another crinoid. This is a non-stalked crinoid, so this would be what we refer to as a feather star. So again, Echinoderm related to the sea stars and the brittle stars. And just earlier we've been looking at a couple of stalked forms. Those are called sea lilies, but all of them together are crinoids. And this one you can see has these little short leg-like structures called cirri that it kind of grabs the rock with. And once again, the mouth is oriented away from the rock, unlike sea stars, and it uses those arms held up in the water column to capture its food. Thank you. You can also see a little cone in there. It looks like a little volcano in the middle. It's a nice sort of theme, isn't it?
I like that.
Seamounts, yeah, volcanoes. That cone is actually, the anus is up on that structure, so it's separated from the mouth so that there's a little bit of separation. So it has kind of a U or a J-shaped gut. So that's also different from sea stars, where the openings to the digestive system are on the opposite sides of the body. So the mouth oriented towards the rock and the anus oriented upwards. Thank you. Good view. So for some of you less familiar with biology, those are animals. They do have a mouth and a gut, have to get their food from the water column. They're capable of sexual reproduction, so they're typically broadcasting sperm out into the water column. The females would take up the sperm, fertilize the eggs, and so on. The heading of our ROV right now is about 320 degrees, so we're kind of pointed towards the northwest as we make our way up a slope that is more or less the overall direction we're going today is north to northeast, I think you would say.
Yeah, that's fair.
Another one of these Brazing sea stars. So it's interesting, we were just looking at the Crinoid. So this is a relative, another kind of Echinoderm. It's a type of sea star. So this one, although it has the same long arms, right now they're not held up into the water column, but normally when they're feeding they would, just like that Crinoid that we saw. But here the mouth is pointed down. And then in front of it you see there's what, a dozen or so of those little box-like structures. We saw that earlier, also associated with one of these Brazing sea stars, and those are all little Tunicates. And the thing behind it that's waving on that stalk is another kind of tunicate. So I don't know if this is a young version of the one that's behind it or if these are two different species. Again, tunicates are in the Eurochordata. Oh, I'm going to stop right there. There's a U-shaped tube just at the lower left of the screen. And my initial guess on that, looks to me like the tube that a tube anemone would build. Basically, anemones and jellyfish and corals, all the cnidarians have these cells that are called cnidae, and you may have heard of nematocysts. Those are the stinging ones. So everybody knows, Portuguese men of war, they sting.
Right.
Well, tube anemones have a special version of those cnidae that they fire, and with mucus they can bind up sediments and they can produce these really beautiful tubes. And they're either down into the mud or where there's a lot of rock here can be coiled up like this, and then they can retract into those tubes. So I suspect that's what it is. I don't know for certain. If it is, it would essentially crumble as soon as we touched it. But certainly I don't see anybody home right now. Maybe. But you can see, you see on the right side here, it's really, really smooth in there. I mean, that's got to be a tube anemone tube. It's almost velvety. And you can, people keep these in their aquaria. They're in shallow water as well.
Oh, really?
Yep. Thank you. That's a nice view.
I noticed there's a lot of life up on this bigger rock here. Are they getting better sense of the currents there? Or,
right. So when you get up off the bottom, when you're right at the bottom, the current is effectively zero. So the more off the bottom you can get, the higher the currents. And a lot of these things are dependent on their food from drifting through the water. So they want to get up onto this kind of topography. And so this is kind of a micro view of the whole seamount concept, right?
Right, yeah.
The seamount is a giant rock sticking out of a muddy plain, and so lots of organisms live on the seamount that don't live on the muddy plain. There's a sponge waving back and forth. And I didn't finish my thought there, the tunicates. So tunicates are Eurochordates. They're basically in the phylum Chordata, which is where the vertebrates are. So those are your closest living relatives that have no backbone, even though they don't look much like you. Gina Molotova does not think it is a tube anemone tube. So that creates some mystery. So we see another Brazing sea star and another one further in the distance. They all have their arms down, which is interesting. I'm very used to seeing them in feeding mode with their arms raised. So there's something red floating through the water column here. There's maybe, oh, it's a shrimp. I believe it's a shrimp swimming head on. Yep. So you can see these shrimp have these extraordinarily long antennae that allows them to detect things at quite some distance from their body. Keep in mind, you know, the D2 ROV has got these huge lights on so we can see everything, but normally it's very dark down here. And so a lot of the detection of prey and food and so on is either using sensory, excuse me, it's all sensory, mechanosensory touch, like maybe with the antennae, or chemosensory smelling. And you can see the swimmerettes or the pleopods. These are the appendages of the abdomen that are gracefully beating there and causing it to swim. Yeah, great. 'Cause I saw we passed over a different kind of coral that we hadn't seen yet on this dive. And it was white. Let me still back up to the left. Oh, maybe it's at the top of the screen now. Yeah, just coming in the top of the screen to the right of that larger rock. There's a white colony. So here I can see as we're zooming in, the holdfast is on the side of this rock. So that would be the place where the larva first settles, kind of glues itself to the bottom, and then it begins to grow and asexually bud new polyps. And you can see that it's kind of curved out from the side and then is going upward again to get into this faster flowing water. So we actually have two things here. So we have a bamboo coral, that's the colony that you see, and this is likely the same one that we saw very early on in the dive. It's got a few branches. And then there's a crinoid, a sea feather star on it. Actually, no, this is a new bamboo coral. This is different. So this is we have not seen this on the dive today, and one of the reasons I can say that so unequivocally is there are no long spines between the tentacles, and I'm seeing lots of it's hard to see through to the skeleton. Earlier I was telling you the tissue is transparent, which means there's probably a lot of scales inside this tissue. So those are good taxonomic characters. Great view, thank you. We can move on. So I think that's the third.
Oh, come on, Scott, take a guess on this one with all those dense sclerites inside of it.
I will take a guess that is Jasonisis. You know, I can't believe I'm so absorbed in this, Chris, I didn't even bother to look to see if that was branching from the nodes or not. There's too much going on here. Did you see?
You couldn't really see, Scott. In fact, I'm not sure I even saw a very recognizable node at all on this one.
Well, you're not as fortunate as I am to have a videographer sitting next to them, and Caitlin has just shown me an incident replay, and it is not branched from the nodes, it appears. But my initial guess is Jasonisis. I would like to hear what Les Watling, who's also at the University of Hawaii and an expert in this group, has to say about that. So the interesting thing is, I think we've seen several corals. I don't believe we've seen more than one of each. It's like every time we see one, it's something new. We have another one of these sort of what I call bulrush sponges. This is the largest one I think we've seen so far today. And Chris, can you remind me what we were calling these? Or what you called it earlier? Or maybe Shirley, I think you said that this was a demo sponge?
Shirley, you want to take it? Okay, I'll do it. Piloderma is what we think.
Sorry, I didn't get to it fast enough. Go ahead.
Shirley, would you agree this might be another one of these Piloderma?
Yeah, Chris, I looked in the systema peripheral, and it looks like it might be.
We also see several of those Tunicates down below that sponge and another Brazingid. So far I think the Brazingids and Tunicates are winning the day in terms of abundance. Oh, that was the one we saw earlier. I apologize because there's the one that's in the background. So we shouldn't double count those. Yes, Nav, I can hear you. It's watch lead. Copy that, Nav. So, ladies and gentlemen, as we troubleshoot an issue here on the ship side, the surface side, we're going to just come off the bottom for a few minutes and just make sure that we have all our controls before we continue moving up this slope. So just stand by.
Can you switch with me, please?